everyone, I'm Asiyah. I'm a Master of Science student at the University of Saskatchewan, and today I'm going to talk a little bit about canola protein extractions. I will start with a brief overview of canola in Canada and then move on to some reasons I think it's important to be looking at improving the utilization of canola, and in turn, why it is important to establish alternative protein sources. I will then talk about the method of protein extraction and that I chose, as well as some facts regarding the resulting protein isolates that I have obtained. I will briefly talk about my expected extraction efficiency as well. So, canola in Canada. Canola is heavily produced in Canada, with the main areas of production falling in the Western Canadian provinces of Saskatchewan, Alberta, and Manitoba. Canada is also a major exporter of canola and the number one producer of it. So to put this into perspective, in 2020, over 20 million acres of land was used to produce canola in Canada by 43,000 farmers producing over 18.7 million tons of canola in Canada alone. Canola is well suited to growth in semi-arid environments like that of the Canadian prairies, but because of the variability in weather and soil quality, the yield, the composition, the oil and protein content are all affected. In my study, I look at 13 samples grown throughout these three Western Canadian provinces that you see in the picture to the right. So while canola oil is the main reason for producing all of this canola meal, a byproduct of the oil extraction process, has drawn a lot of attention due to its nutritional profile and its functional characteristics, as well as its discernibly high meal protein content. So, in the sense of sustainability and moving towards the future, there is a growing interest in the utilization of canola. Canola proteins have a well-balanced amino acid profile and has more sulfur-containing amino acids and lysine than both cereals and pulses, which makes it a good candidate for both animal and human nutrition. Looking at the fact that the world's population is expected to grow to 10 billion people by 2050, that is 2.1 billion more mouths to feed in a very short period of time, it is essential to maximize the utilization of what is already being produced, especially underutilized high-protein oilseeds that are already mass-produced, such as canola. Canola meal has 40% crude protein value on average. With protein being the limiting nutrient and the demand for good quality protein increasing, as well as changes in sociodemographic characteristics of the current growing population, it makes sense to try to maximize the use of what is already being produced. Which brings me to my project. The impact of regional variability on the nutritional profile and the functional characteristics of canola protein isolates. As I mentioned before, I have 13 samples spanning the Western Canadian provinces. I look at 2 strains grown in Peace River, Alberta. I look at 5 strains grown in Vermilion, Alberta, another 2 strains in Colonsay, Saskatchewan, not too far from here, and another 4 strains grown in Crystal City, Manitoba. So the samples portray a wide variety of canola grown over a very large region, which encompasses the area where 99% of the canola is grown in Canada. So what you're seeing right now is the average crude protein percent based on strain of the seed samples that I do have. This is a table I drew up which basically shows you that the range of the seed samples I have is between 17 to 26%, and with Colonsay having the most crude protein at 26%, and Peace River having the least at 17%. The average crude protein content of seed samples is around 21.9%, and generally canola seeds have a crude protein content of approximately 20%. So it's a good variety, some below, some under, and some right around the average. When you look at this based on location, the chart below gives you an idea of the average protein content based on region. Part of my project looks at the regional variability, and I think this depicts it well. It'll be nice to see how this plays out when all the data is in and we can look at how this compares to the protein samples and the average crude protein and the protein isolates. So given that I had seed samples, I used a continuous screw expeller press, seen in this picture, to extract oil from the seeds. I then used countercurrent extraction by submerging 50 grams of the pelleted seed cake that you see in the silver metal bowl into 4 separate beakers filled with 1 liter of hexane. They were in each beaker for 15 minutes in a stepwise manner, and all samples went into hexane samples from dirty to fresh. This is explained, as you can see in this picture, it kind of explains that a little better. It shows 4 beakers of hexane, and after each sample is submerged, the number 1 beaker, the one without the foil covering it in the picture here, is discarded. The beakers are then moved to the left so that the last beaker, beaker number 4, always has 1 liter of fresh hexane in it for a sample to go in. This can be seen in this picture. The 3 dirty beakers have been moved to the left and have become beaker number 1, 2, and 3 from dirtier to cleaner, and the 4th beaker is always fresh hexane. And this is where the sample will spend its last 15 minutes. The defatted meal is then dried under a fume hood for 24 hours and subsequently toasted in the oven at 100 degrees Celsius for 30 minutes. You can see this in this picture here at the back of the fume hood. There's those 3 aluminum foil trays, and they have that canola meal in them that's drying. So when we're looking at the average crude protein of canola meal based on strains, the canola meal that I obtained from this process, this table gives us a good example of that. The protein content of canola meal obtained is seen in this table. If you look at the total, like the average of the canola meal samples in the table, it's 39.8%, and it ranges between 33.7% and 45.2%. Generally speaking, canola has a meal protein value of about 40%, so again, just like the seed samples, it's a good range. They vary slightly, with some being above average and some being slightly below. Take strain A, for example. In Collins A, strain A has a 45% protein content, but in Crystal City, about an 8-hour drive away, it has a 41% protein content crude value. So if you're looking at Vermilion and Peace River, which are both in Alberta, but in separate soil zones, you're seeing a 6% drop in protein content as well. When looked at based on location, you can see the effect of growing location on protein content, which this graph shows. So now going back to the extraction method, the canola meal is then stirred with an alkaline solution. So I use sodium hydroxide for 20 minutes and then centrifuged it at 8,070 RPM for 30 minutes. The byproduct obtained was weighed and placed in the oven for drying at 100 degrees Celsius for 24 hours, after which it was ground and placed in the fridge for further analysis. The supernatant was titrated to a pH of 4.5, after which it was recentrifuged at 8,070 RPM for 30 minutes. The precipitate protein extract was then freeze-dried to obtain a protein isolate. The liquid portion was dialyzed using 6 to 8 kD dialysis tubing in 100 volumes of distilled water overnight, after which it was spray-dried to obtain the soluble protein portions. So unfortunately, I'm still in the middle of my extractions at this point, but this means that I don't have too much data on the protein isolates available to show at this point. I do have some preliminary data from the first 2 replicates that have been completed, and so I'm going to talk about that a little bit right now. This table shows the protein content of the precipitated protein isolates that were freeze-dried, and the average is based on duplicates currently rather than the quadruplicates it will be based on at the end of the study. As you can see, the average crude protein percent for freeze-dried samples is currently 76.1%, but it ranges from 68.5 to 80.3. Now this is quite a wide range. The low percent protein in sample 4 can be explained in part by an error that was made during extraction process. So sample 4 replicate 2 was titrated with 1 molar HCl instead of the 0.1 molar HCl that was used in all the other samples. So because of the higher concentration, the pH dropped quite quickly and well beyond the endpoint of 4.5, which can explain the difference between the sample 4 replicates. This next slide gives you an example of that. It kind of shows the factors. In Rep 1 of sample 4, strain D, the average crude protein in the freeze-dried sample was 71.3%, and 9 grams was obtained. While in replicate 2, the sample that used the 1 molar HCl instead of the 0.1 molar HCl, the percent protein was 65.7, so that's quite a bit lower. So the spray-dried soluble protein content, looking at that, you can see that on the table here, I have most of the samples for Vermilion completed, and I have 2 of 4 for Crystal City, 1 of 2 for Colonsay, and 1 of 2 for Peace River. The green boxes basically just show strains that I don't have in those growing regions. So the spray dried soluble content as of now is looking like the average protein content is around 75%, and the range is currently between 80.6% and 72.2%. I'm expecting to see an extraction efficiency of about 55 to 60% based on what I have seen right now with the samples that are completed. I also expect to see an apparent difference in protein content based on location, with Peace River having the least percent protein throughout, and Colonsay, Crystal City, as well as Vermilion having the most, depending on the strain we're looking at and the location. And I do look forward to testing the functionality of these protein isolates and determining how functionalities vary by region, if they vary at all. Thank you for your time, and I hope you enjoyed my presentation. The next few slides are my references for this presentation. Thank you.